For this presentation, I am going to explain how carbidopa can be used to treat the symptoms of Parkinson's disease. I'm also going to talk about a little bit about the process of X-ray crystallography and show the bound structure of carbidopa. The first topic is carbidopa. Levodopa is the combined formulation is called Sinemet. It's the most effective drug to treat Parkinson's disease. It's delivered in many forms: immediate release, slow release, gel form, and through an inhaler. It's the most effective medication available for Parkinson's disease. It treats the symptoms. It does not change the progression of Parkinson's disease. Carbidopa. Carbidopa enhances the effect of levodopa treatment. The lower doses of levodopa can be given when carbidopa is added. It prevents the conversion of levodopa to dopamine. It reduces levodopa-induced nausea and vomiting. Use of levodopa to stop it from breaking down before it crosses into the brain. It has no effect when it's used alone. It is in a class of medications called decarboxylase inhibitors. Parkinson's disease. Parkinson's disease is basically a loss of neurons in the brain that use dopamine to communicate. It causes slowness, stiffness, tremors, and balance problems. It causes bradykinesia, postural instability. Cells that make dopamine are impaired. As PD or Parkinson's disease progresses, more dopamine-producing brain cells die. Levodopa is a chemical found naturally in our brains. It's transported to the nerve cells in the brain that produce dopamine when used as a medicine. It can be converted into dopamine for the nerve cells to use as a neurotransmitter in a case of medications called central nervous system agents. The side effects of carbidopa/levodopa therapy are nausea, vomiting, orthostatic hypotension, worsening of glaucoma, dyskinesia, hallucinations, psychosis, low blood pressure, dry mouth, and dizziness. Carbidopa and levodopa therapy is a combination medicine. It's used to treat Parkinson's symptoms caused by carbon monoxide poisoning or manganese intoxication, along with slowing the progression of or slowing down Parkinson's. It may be used for purposes other than Parkinson's disease. X-ray crystallography uses electromagnetic radiation to determine the molecular and atomic structure of a crystal. The crystal structure causes X-rays to diffract in specific directions. A 3D picture of the electron densities are produced. There are 4 steps to X-ray crystallography. The first step is protein crystallization. The second step is production of a diffraction pattern. The third step is analysis of the diffraction pattern to produce an electron density map. And the fourth step is determination of the protein structure. The crystal structure must be flawless to have the position of the electrons mapped accurately. This is a picture I found that kind of diagrams X-ray crystallography. It starts with the crystal, then you have the X-rays with the crystal, causes the diffraction pattern. It phases into the electron density map, then it fits into the atomic model. Those are all like the parts of refinement. It kind of goes from the crystals down to the model in the end. Here's some of my references. A few of them. And then I have a picture of the bound structure of carbidopa. So I'm going to get out of my PowerPoint to show this is the bound structure of carbidopa. It was from the Chimera model. And that is it. Thank you.